Hi, everyone. Welcome to the Brain Foundation webinar on Neuromodin for Autism. I'm Sarika Agrawal, your host for today, and also the co-founder of the Brain Foundation. And joining me today are the team of PoloMar Health and leading autism physicians, whom most of you probably know of, who will be participating in the upcoming trial for Neuromodin. The genesis of this effort began earlier in this year when PoloMar Health approached us to partner for this effort. For those of you who don't know about Brain Foundation or the work we do, I'd like to tell you a little bit about why this was very exciting for us and why we are joining PoloMar Health to make this effort happen. So I'm going to just walk you through a little bit about the Brain Foundation and why this work aligns with our mission, and hopefully we'll get into the discussion on Neuromodin. The primary mission at the Brain Foundation is to catalyze research that can lead to the development for FDA-approved treatments for disabling symptoms of autism and neurodevelopmental disorders. Now, this is something that I probably don't have to explain to all of you. Many of you who are joining understand why this would be a very important mission. Many of you are on this journey for a while, and we hear all the time from families how much they're looking for answers and more evidence-based medicine. So there are so many other foundations and various governmental organizations that are also supporting research that can lead to FDA-approved treatments. We believe that we are very unique in how and what we are doing to help get to this outcome. Number one is we are a complete community-directed organization. We are for the community, we are by the community, we take community inputs constantly every year to establish our research priorities. We also focus more on translational research. It's one of the less invested areas, but we believe focus on translational research is what's really needed to get to faster outcomes than we have seen so far in over 20 years since autism research really started happening. The other thing that we want to emphasize is prioritizing subgroups that have historically not been as involved in this kind of research. They have been overlooked. These are children who are minimally speaking, non-speaking adults, those with very high support needs. We also are unique in that we not only support research happening in the academia, but we also partner and support biopharma companies. And last but not the least, we are a team, our board, all the folks that work in our foundation, everyone has a loved one with autism. So we really have a very deep and personal stake in this. So far in the 3 years of our founding, we are a relatively new organization. We've funded over $2.5 million in research grants. Areas of funding have included biomarkers, some basic science, using animal models to study metabolism, organoids, mitochondrial dysfunction, immunology, etc. And these are just some examples. Please go to our website at brainfoundation.org to take a look at all the past research grants that were funded by the Brain Foundation in the last 3 years. We also make highly selective equity investments in very, very promising startups through venture philanthropy. A key part of our work every year is our flagship Synchrony Symposium. This is the first and only translational research symposium you're going to find anywhere in the world where people from academia, people from pharmaceutical companies, members of our community, venture partners, all collect in one place to intermingle, to exchange ideas, to learn about each other's work, to make new connections. And we do think tanks and roundtables. We also obviously have presentations, and we have a lot of fun as well planned. So if you haven't been part of Synchrony before, I encourage you all to join our upcoming Synchrony Symposium in October. That'll be happening in California. We also work with many biopharma companies, and today our focus is on PoloMar Health, that's developing Neuromodin. And I'd like to then take this opportunity to invite Dr. Ashley Jordan from PoloMar Health to tell a little bit about the company, to tell us about Neuromodin and the upcoming trial. So with that, let me stop sharing my screen. And Dr. Jordan, please take it away.
Thank you for that introduction. So I will be sharing a presentation. Let me just pull that up. Sarika, are you able to give me control?
Yes, should be. Yeah, there should be. You should have the control.
All right, just pull this up. Okay, is everyone able to see the screen? I'm just gonna pull up the presentation. All right, everyone should be able to see the screen now. So hello, everyone. Thank you for your time and attending this webinar. Again, just to echo Sarika's previous comments, we will be talking about a clinical trial as well as a new supplemental product that will be used in a clinical trial investigation, PoloMar Health. So just to get started, PoloMar Health was established in 2022, and the main mission that the company has is to help patients and physicians with unmet medical needs identify and access therapies that are not currently approved in the US. And so in order to do that, we have to coordinate and manage the complicated process that's involved with accessing these medications. And so just to kind of get into the science behind the rationale for what we'll be discussing today, again, we won't be necessarily going into in-depth with the symptoms, as many of you are well aware, but we will be talking about some of the basic science behind the rationale for the new product, as well as the clinical trial that we'll be discussing today. So within ASD, there are several molecular mechanisms that have been studied and researched that may, attribute to the etiology of the disorder. So this slide just kind of shows how all those different pathways kind of lead to ASD symptoms. And the two that we'll be discussing today include nuclear factor erythroid as well as purinergic signaling. And so to just kind of dive right into it, purinergic signaling is a form of extracellular signaling. So basically, within a cell, these signals help to alert the cell to stressors, external stressors or internal stressors. And once that occurs, the cell can then decide to upregulate other pathways that help the cell to deal with that stress. And within ASD, specifically, we're talking about, or we're looking at these receptors, the P2RX7. So what happens is the purine comes in and kind of signals, upregulates this pathway, and you have a cell danger response. And this is one of the mechanisms that is thought to attribute to or contribute to ASD symptoms. In addition, we'll also be looking at NRF2 signaling, and that is just another signaling cascade that is involved with dealing with oxidative stress. And that is another form of stress, cellular stress, that has been found to contribute to, again, ASD symptoms. And within ASD, these two pathways are dysregulated and will be the focus of the talk as we go on. So to kind of put everything all together, preclinical research has shown that a potential root cause for ASD may be this persistent cellular stress that occurs, as well as the dysregulation in the two pathways that I just mentioned. And so what happens is, in general, there is a perception of cellular stress. So the cell is under some form of stress. The cell danger response is triggered, but along the pathway, you have some abnormal response to the cellular stress. This leads to persistent cellular stress, which then can lead to ASD symptoms. And again, this is not a comprehensive synopsis. There are many pathways that have been found to attribute to ASD, but again, we're kind of just focusing on these two because they support the rationale for the clinical trial that we will be discussing. And so clinical studies that target these pathways, we are in hopes that they will lead to a safe and effective therapy for ASD patients in the future. So to begin our discussion on Neuromodin, which is a dietary supplement that will be included in the clinical trial that we'll be discussing. So briefly, Neuromodin is a natural extract blend of Emodin and another additive known as Piperine, which we will be discussing. Neuromodin is a highly bioavailable proprietary form of Emodin, and it can be extracted from the roots of plants such as rhubarb and some other plants as well. And here we just have a picture of what the molecule looks like. So some of the systemic effects that Emodin has been found to contribute to are anti-inflammatory, neuroprotective, as well as anti-diabetic. So Emodin is a compound that does have a fair amount of research behind it as to how it can affect the body. And again, just going a little bit deeper into the neuroprotective effects, the Emodin has been shown to reduce neuronal oxidative stress, as well as neuronal cell death, and also reduce neuroinflammation. All three of these have been associated through research, through studies with ASD as well. And so to kind of tie everything together, the reason why we kind of went through those first two pathways of how ASD symptoms may be occurring. That first one we spoke about was purinergic signaling. Essentially, this receptor is activated by this purine ATP, and you have the cell danger response, which leads to negative effects within the cell, excuse me. So one of the hypotheses behind how Emodin may be acting is through an anti-purinergic pathway, where essentially the purine signaling is blocked. Therefore, the cell can then deal with the cellular stress that is occurring in the context of ASD. Similarly, with NRF2, you have this, this degradation of NRF2, which can lead to cellular damage. Again, all of this is happening in the presence of cellular stress in the context of ASD. And one of the other mechanisms of action that Emodin has been shown to have is cytoprotective. So Emodin is able to increase NRF2 levels, which then allow the cell again to signal other pathways that help the cell to deal with the cellular stress that is occurring. So just a bit about the pharmacokinetics. As I mentioned, we won't go too deep, but we did want to provide the rationale for why Emodin is such a molecule of interest. So this particular study kind of supports the combination of Emodin and Piperine, which is that additive that I mentioned that's included in the Neuromodin supplement. Essentially, this study looked at the bioavailability of Emodin. So how much of the Emodin is able to actually get into the body and get to the places where it needs to go in order to affect some therapeutic change. And so in this study, mice were given either Emodin or Emodin plus Piperine. and they wanted to kind of compare some of the metrics that are used to determine how well the substance is absorbed by the body. So what they noticed was for bioavailability, plasma concentration, which is just peak plasma concentration, which is the highest level that the substance is able to achieve within the body, as well as overall plasma concentration. All of these metrics were significantly increased, as you can see here, with the addition of Piperine to Emodin. So again, we're just kind of showing the rationale for why Emodin and Piperine are being included together. Also, it's very, very important for us to look at the tolerability and safety of Emodin. So these are two studies that kind of looked at those metrics as well. This first study, there was a blood sample taken from a healthy patient, and essentially those cells, those blood cells, were exposed to Emodin. And what the researchers did was they looked at the cell viability. So how many cells essentially are able to survive after Emodin treatment. And what they found was at these concentrations, Emodin was found to be safe. Now, at these higher concentrations, they did see a slight dip in cell viability. But again, these are concentrations that aren't necessarily therapeutically relevant, concentrations that may not be achieved within an actual living organism. So overall, what they concluded is that Emodin is like safe at low levels, but may have some negative effects at very high levels. Now, the second study that you see on the right, this was a study done in live organisms. So male and female mice were given a standard diet of Emodin mixed in with their food. And essentially what they found was there were no toxic effects seen within the liver, which is shown here. They also looked at the kidneys as well as the heart, which is not shown. But again, the major conclusion here is that the mice given the Emodin, they didn't see very many toxic effects at all. So they concluded also that Emodin was generally safe. So just to summarize this section, Emodin is a naturally derived active ingredient of Neuromodin. Studies have indicated that Emodin has several neuroprotective effects, and that the proposed mechanism of action for Emodin, in some, there is mechanism of action studies as well as safety and tolerability studies for Emodin. And those two main pathways that we think Emodin may be acting through are NRF2 as well as purinergic signaling. And finally, Emodin may address signaling pathways that lead to ASD symptoms. However, more studies are needed in order to determine the efficacy and safety in humans. So just getting into the PaxMedica study, which again, we're talking about the anti-purinergic aspect of addressing ASD symptoms. So essentially, investigators were interested in determining the effects of anti-purinergic therapies, such as Emodin, in a mouse model. And this mouse model is a mouse model for something called Fragile X syndrome, which many of you may be aware of. Fragile X syndrome is typically associated, or it's generally associated with ASD in rates of 16 to 46%, up to 46% in some patients. And essentially this mouse model is a proxy for being able to study how a compound or how a therapy would affect someone who also has ASD. So within these mice, there were 3 tests that were done. The first was passive avoidance, the second, social novelty, and third was the Morris water maze. And essentially, we have, some pictures to kind of describe visually what these experiments are testing. But essentially, mouse is trained to fear the dark, which is against their natural inclination. And this test is meant to assess knowledge retention. The social novelty test, within the social novelty test, a mouse is introduced to another mouse. They become acclimated, and then they're introduced to a second mouse who is then considered a stranger. And so this test is meant to assess the desire for socialization. Now, this third test basically is a test of spatial and short-term memory, where mice they have to find a platform within a shallow pool of water. And the quicker they learn this path, the quicker they can get to the platform. It kind of assesses or shows that their spatial and short-term memory are improving. And what the PaxMedica APT study showed was that mice treated with Emodin were able to basically excel at all three of these tests. Emodin treated mice spent 100% of their time in the lighted box, indicating that they had increased knowledge retention. They spent more time with the strangers that they were introduced to, indicating an increased desire for socialization. And they were also able to find the platform and reduce their time in comparison to mice who had not been treated with Emodin. So again, Emodin is kind of increasing all three of these measures. And all three of these measures are associated with ASD. So in summary, the FMR1 knockout mice, again, this is a mouse model for Fragile X, is also used to kind of study some of the symptoms of ASD. They were treated with Emodin, and all of them were able to increase their general knowledge retention, their sociability, as well as their spatial memory. And the results of this study is somewhat of the basis for the study that we're going to talk about with Emodin in the clinical trial, looking at the effects therapeutic effects that may be seen with ASD patients. And so moving into specifically talking about the Neuromodin clinical trial. So PoloMar has designed a trial that will test the effects that Emodin and Piperine together will be able to have on ASD patients. Essentially, we are looking to enroll between 40 and 48 patients. Of those patients, they will be randomized into 4 different groups, depicted here, A through D. And those groups will have different amounts of Neuromodin given to them over a 12-week period. Now, within each group, the hope is to enroll between 10 to 12 patients who will each receive these varying concentrations of Neuromodin that you see here, 40 milligrams, 200, and 400. And then there'll be a placebo group to serve as a control. And so that dosing will occur for 12 weeks. And then there will be a 6-week follow-up. So the abbreviated procedure includes the following. Investigators will determine the correct dosage, and that will just be based on their assessment, also based on the body weight of the patient. 1 scoop of Neuromodin powder will be mixed in 8 ounces of water for every 20 kilograms or 44 pounds of body weight. The entire preparation would need to be consumed within 1 hour, and then that total preparation would be consumed daily. And if it wasn't clear, the formulation is in the form of a powder. So this will be a powder mixed into a beverage. So the final step would be patients will maintain follow-up visits per the final protocol schedule. And again, these are things that the investigator would be letting the patient know throughout the trial. So in terms of some of the endpoints, I won't read all of the bullets. Again, it's a very text-heavy slide, but this is just to kind of show what the investigators will be looking for throughout the study. So some of the different scales, they'll be patient assessed as well as caregiver assessed. So some will be assessed at, or all will be assessed at baseline actually. Some will be assessed at week 6, and then there will be endpoints assessed at week 12. So in terms of the safety endpoints, any adverse effects that occur during the trial will be noted and will be addressed accordingly. And again, that is up to the discretion of the investigators. As you can see, we've also included some of the inclusion criteria here. I won't read every bullet, but essentially, the trial will include children ages 5 to 12, both male and female children. And there's different inclusion criteria that will allow the patients to be eligible for the study. And with inclusion criteria, we also have exclusion criteria listed here. And again, I believe these will be made available. Zach or Addie, let me know if that's incorrect, but exclusion criteria will be made available. And again, your physician or the investigator will, assess whether or not your child is eligible for the study. And again, this is just the final list or the rest of the list of exclusion criteria listed here. So as many of you may be wondering, how can patients join the trial? There is a link, so you can go to the Brain Foundation website, and there you will find a form that you are able to fill out to express your interest. And within that form, they will collect all the information they need from you in order to kind of assess the initial eligibility. And some of the frequently asked questions that we've been getting, from people or caregivers and children who are interested in the trial, what are the location sites? So there's 3 locations, Naperville, Illinois, as well as Foster City, California, and Wilmington, Delaware. In terms of whether your child needs to be a current patient of an investigating physician, investigators will assess the eligibility of all interested participants regardless of their current patient status. So you do not necessarily have to be a patient of one of the investigating physicians. And when, in terms of the timeline of when you'll find out whether your child is selected, that will be about 3 to 6 weeks after you submit your form. So it's kind of the turnaround time for when you would hear back from the investigators. So at this time, we'll take any additional questions. Again, thank you for your time. I hope this provided some clarity on the background of the clinical trial and what PoloMar is hoping to achieve.
Thank you, Dr. Jordan, for that very informative presentation on Neuromodin. And I can see a bunch of questions coming in from our audience that are very excited, obviously, at the idea of a new product being available. Before we go to questions, I'd actually like to give an opportunity to the physicians that have joined this effort. Many of you know them, but let me still introduce them. We have Dr. Anju Usman Singh from True Health Medical Center. We have Dr. Dave Fliedner from Lighthouse Complex Care, and we have Dr. David Traver from his private practice in Foster City, California. And we have Dr. Rossignol as well here, maybe from Florida or California. I don't know where, but I'd like to open the forum up to our physicians. Maybe if you can each take a minute or two to talk about why you're interested in and why you signed up to participate in this effort. So Dr. Singh, if you want to unmute yourself, please, you'll be the first one to take the floor.
Sure. Hi, everybody, and thank you to the Brain Foundation and PoloMar for doing this. I've been working with children on the spectrum for almost 30 years. I know I don't look like it, but I have been. And I just, we've been trying all kinds of things to help our patients, from diet and supplements, and trying to leave a legacy where we are actually getting a study done on one of these very promising supplements. And the whole idea of the knotweed and the Emodin and the Piperine, NRF2 signaling, purinergic signaling is really exciting. And so I know my patients are excited about this, and I'm very excited about this. So thank you.
Thank you, Dr. Singh. Dr. Fliedner, any words you'd like to share?
Yeah, thank you. I want to repeat the thank you to everybody involved, and we're really excited to be part of this study. We're opening up at Lighthouse Care in Delaware starting in mid-July, and that's going to be the site of our clinic. I think many people are familiar with us out of Denver, Colorado, but we have a bricks and mortar opening in Delaware, so I'm excited to really represent, I guess, the East Coast in a sense, and kind of that tri-state area where there's such a huge concentration of need. Similar, I've been in pediatrics for over 20 years, and I've always been frustrated by my inability, or kind of our collective inability, to really help a lot of these kids. And like Dr. Singh, I've tried many, many remedies, and sometimes they're successful and sometimes they're not. And so I think we've all heard about the pathway and been frustrated by our inability to really affect that change with the cell danger response as much as we would like to. And so I'm really excited to be able to have something that maybe is going to be a possible remedy to try to help mitigate that problem. So we're really super excited to be part of it, and we really want to thank everybody for letting us be involved.
Thank you, Dr. Fliedner. Yeah, we're very excited that you and the team of Lighthouse is participating. Dr. Traver.
Hi, everybody. And I echo the gratitude of my colleagues. It's a privilege to be here, and not just to be part of today, but I think we all recognize that the relevance of this study goes beyond a webinar. It really resonates from some of the research that's been done on similar products, similar compounds, I'll use that word, showing that, giving drugs the tradition of the current day science that targets some kind of monomechanistic effect is just not adequate. And I think we have an unhealthy over-reliance, I think very evident, on agents that produce marginal benefit and unfortunately significant side effects. This is not lost on the audience, I'm sure. And so, like Dr. Fliedner and Dr. Singh, I'm very grateful because for the past, again, 2 to 3 decades, I've been working, and I echo what Dr. Singh has shared. It's been extremely frustrating. At the same time, we know that the answers are out there. We believe that there's work to be done. And for that reason, I think that when we recognize the diverse array of metabolic and immune conditions that these set of conditions exhibit, we really need compounds which, from a foundational level, can address this spectrum of abnormalities. And I think this is one such compound. So I'm grateful to be here, and I look forward to hopefully participating.
Absolutely right on, Dr. Traver. And I just want to echo the same thing. This huge unmet need that we have out there is the reason why the Brain Foundation was formed, why we are funding research like this and much more. So yeah, absolutely. I think our community cannot wait to see better answers available sometime in the future. Dr. Rossignol, I'd like you to join us and say anything that you'd like to add.
Yeah, so my name is Dan Rossignol. I have 2 children with autism. Been treating kids with autism for about 20 years. I tell people I got dragged into autism kicking and screaming because of my 2 kids. So obviously I became very interested in research. I've been a primary investigator on 3 different studies using HPOT and autism. In one of the studies, we had 6 sites, and I was the principal investigator, so I had to kind of oversee all that. And so that was quite a challenge. I'm also been a medical monitor for 2 of Dr. Fry's study on leucovorin. And so I'm very interested in research. So I'm going to be the medical monitor for the study, which means I'm going to be watching out for the safety, integrity of the study, answering questions from investigators, regulators, and so forth. So my goal is to make sure the participants are safe, that the study is done properly. So mainly on that side is where I'm going to be focused. Any questions and problems in the studies, obviously I'll be helping to oversee all that. But I really appreciate the opportunity to be involved. I'm definitely interested in as much research as we can do to find answers for our kids so we can have more treatments available. So whether this works or not, it's good to know. it's good to study. And I'm excited that this is actually being studied as opposed to just releasing on the market and not know if it works or not. So I think the pharmaceutical companies should definitely be encouraged to do the study so we know if it works or not. So I think it's a great study to be involved with. So thank you very much for thinking of me.
Sure. Thank you, everyone. And gosh, we are flooded with questions here, and I am attempting to figure out a way to sort through this. Regretfully, we will not be able to go through, I think, in the time we have available through all the questions. We'll try our best. Let's get started one by one, and let's see how far we go. One question, and please, all panelists, please feel free to chime in. I'll take one question. The first question, if this is a supplement, is there any chance this product will be available for purchase outside of the trial in the event that we are not able to participate? You want to take this
question?
I'm sorry?
Zach, would you like to take this question?
Yeah, sorry, I'm just trying to answer some of these ones coming in the Q&A. Yeah, so it is our full intention to make the product available for purchase shortly following the conclusion of the trial, and we do intend to make it available globally, and so it it's hopefully only a matter of a few months, and once we wrap up the trial, it will be available and capable of being purchased globally.
Okay, cool. We have audience members who have upvoted this question, so I'm going to take this next. And this is about, there were multiple questions on the effect of Emodin on cortisol. A question says, Emodin looks like it can be used to lower cortisol. Does this mean it's contraindicated in folks with low cortisol levels? Is this pre-measured before people enrolling in the trial?
I can briefly comment. Yeah, I mean, I can maybe just comment briefly. I don't know that, this is something that we have a exact answer to. I think this is likely going to be dependent on a case-by-case basis and a conversation with the physician that is overseeing you through the trial, as far as if they have concerns relative to that or not. So I would say that if this is of a concern to you as a patient or a physician, or a physician having a patient come to you, I would maybe just take a close look and have a conversation as far as if this is something that you feel comfortable with. Certainly there's certain research around Emodin's effects with cortisol, but I don't know necessarily if it's understood well enough to have any blanket statement as such. So that would be my suggestion, and if any of the physicians would like to comment on that, that'd be great.
I'll comment on it really quickly. We're thinking about the cell danger response, and we're thinking about how the adrenals affect on that. And oftentimes we will see high cortisol, but some of these patients who have been sick for so long, the cortisol might drop. Most of these types of natural substances have adrenal modulating effects, and they're not so simple as lower or higher in cortisol. They do modulate cortisol. So in general, my experience with substances like this, it hasn't had a negative effect on people with low cortisol. In fact, the opposite. And we're not going to be doing any laboratory testing, so it wouldn't be something that we would actually know ahead of time for most of our patients who are doing the study.
Thank you, Dr. Singh. And I'm so happy in a way to see these questions coming in. People are commenting about the age of the children that will be included in the trial. And right now, this current trial restricts or is limited to children who are age 5 to 12. And we have a number of questions saying, why not adults? Will there be a study on the adults? So if you can comment first on how this age group was chosen, and then I'd like to add about, my personal commentary on what we want to do to make this trial available for adults as well.
I could comment just briefly again, and then again, very happy to have anyone else chime in. You know, the age group, we did have a lot of conversation and thought with respect to this age group. You know, originally it was a bit broader, but, you know, speaking with the physician experts, it was really intended to try and have a homogeneous population so that we can get as best understanding of the data as we can. And to do that, it was suggested that we stay within this relatively narrow age range, because, you know, broadening the age range, of course, just introduces maybe additional variables that have to be parsed out from the data. So it was really in an attempt to get a clean data set that we can at least understand some initial pieces around the product. And then certainly there's an intention and expectation on our end in the future to do further research and expand upon those understandings.
Yeah, and Zach, with your permission, I'd like to kind of give a little bit preview on the offline discussions we've had, because as I said earlier, one of our mission at the, you know, not one, the, you know, a key priority for us at the Brain Foundation is to make opportunities like this be available, be tested for these neglected subtypes, which includes minimally speaking, which includes adults. And obviously we understand that the very important factor here is to take the fastest path to approval in whatever sub-segment that, you know, it can be, and to make sure that there are no confounding issues, which obviously, as many of us parents know, you know, teen years can present some confounding issues, and the range of 5 to 12 therefore does make sense to us. However, PoloMar understands this very important priority, and because they are a partner of ours, they have agreed to consider a trial if with support from the Brain Foundation in some of these other subtypes that are currently not included in this trial. So I urge you all who are really interested in making this kind of product be available or be tested through a trial to support we will be fundraising for this effort soon. So please join us, please support us in making this second follow-up trial possible, if depending on what the results of the first one show us.
And Sarika, maybe I could just make one follow-on comment to that.
Yes.
You know, this is not a drug in the sense of, you know, needing to go through the standard drug development process of phase 1, 2, 3 until approval. You know, we, as mentioned, will be making this product available immediately following this study. So for the individuals outside of the inclusion-exclusion criteria of this specific study, they'll at least also be able to access the supplement as well, whether that's in a controlled setting like a clinical trial or not. So certainly we, you know, have an intention to further understand the product better, but it's also not the case like a clinical trial for a prescription drug, where if you don't meet the inclusion-exclusion criteria, you have to wait another year, another 2 years until another trial comes, and then see if you're, you know, fit in that inclusion-exclusion criteria. The product will be available for all of the population following this study. So hopefully there's some solace in that difference in this type of product than prescription pharmaceutical product.
Sure. Thanks for that clarification, Zach. We also have questions, people asking, when will this product likely be available?
That somewhat goes hand in hand with the other questions. You know, it'll be available really immediately following the conclusion of this trial. So it's not that we can say an exact date, because we of course don't know how quickly this trial will enroll, and but we do have a hope that this enrolls quickly, and that it's really only a matter of really a handful of weeks where it should hopefully be available if we can enroll this trial quickly and conclude this trial over the course of the next you know, few months.
All right, there's a question asking, what would be the duration of the study?
The study is a 12-week study followed by a 6 weeks of follow-up in which there will be no visits or anything like that.
All right. There's a question again on people outside of those locations, the 3 locations you mentioned. Can they participate or not? Can you talk a little bit about location specificity for this trial?
Yeah, I could comment on that. The trial is open to all participants. We do have a limited number of spaces available for the trial. So it is open to people, really within the United States, all over. However, there is going to need to be some dependence on ability to reimburse some reasonable travel expenses. So we will reimburse certain travel expenses, but it's going to be somewhat dependent, I think, on the process of enrollment. But it is open to all individuals that would like to participate. And the suggestion on our end would be, if you're interested, to reach out to the physician, either your physician or the physician that may be geographically closest to you of the physicians on the call today, and try and get in line to participate. And then, once enrollment begins, we'll hopefully be able to get everybody in that wants to participate.
All right. There's a question on the selection criteria. Do we need both or either of the DSM-5 diagnosis and or a confirmation with the ADOS scale? I think this question comes because most parents that they're aware of, they certainly get the DSM-5, but aren't aware as much of the ADOS scale. So could you just comment on the inclusion criteria related to that?
So the study does require both the diagnosis with DSM-5 as well as confirmation with the ADOS. This is something that you can discuss with your physician or one of the investigators if an ADOS is not something you've completed in the past, and then you can be further evaluated for eligibility in the study.
Right. So in other words, go ahead and apply to participate in the trial if you just had the DSM-5 diagnosis, did not have ADOS, and the information will be shared with the investigators, and they will be able to follow up on that with you if selected. We have a question on some of the science here. Hi, Zach. Emodin and Suramin is supposed to act as a nitric oxide synthase inhibitor. Do you think this could be an alternative explanation for the effectiveness of Emodin or Suramin?
So, as this was somewhat directed at me, maybe I'll briefly comment, but then just make the general comment. I'm not the one best suited to speak to some of the nuanced mechanistic potential of the compounds, whether it's Suramin or Emodin. I will say, just generally, this is really early stage science, so I think there's some understanding of the potential mechanisms of these compounds and some hypotheses as far as how they work. Obviously, you've heard a few of them today, and I'm sure there's others that are out there. I think the answer to your question is yes, it could potentially be an alternative. I don't think anybody can definitively say exactly what's going on a cellular level. I personally am much more of an empiricist in the sense that the reason that we are advancing this is really not based on mechanistic science, but based on empirical science, in the sense that, actually a few years ago, we ran a number of in vivo animal studies in FMR1 knockout mouse models, models of autism and Fragile X. And the reason Emodin is being developed is that it performed significantly better than several other compounds that were tested in that battery of studies. So for me, personally speaking, and I'm just speaking personally again, I can't say definitively on a mechanistic or cellular level exactly what's going on. Certainly we have hypotheses. To me, what's most interesting is simply seeing the difference in response of the animals, in this case, that were treated with Emodin, Piperine, relative to other compounds, and the data are actually quite compelling. So that's what's gotten at least me personally most excited about this. Ashley, I don't know, Ashley or Addie, if you have anything that you'd like to add on a more scientific basis, or if any of the other physicians have any comments relative to that either.
Just to echo what you just mentioned, Zach, mechanistically it is it's not clear exactly what is going on, but I will say that the the nitric oxide or nitric, the one you just mentioned, is a target of NRF2. So NRF2 is one of the overarching pathways that is, can cause those mechanisms to occur. So there is some overlap there, but again, it is more so big picture, kind of looking at what Emodin and compounds like Sermin are able to achieve in the animal studies, which is leading us to want to further investigate them in humans.
Yeah, and as a scientist, you want to know mechanism of action, but as a parent, you just want to know if it works. So
right.
And the truth is, there are many, many, many drugs that are fully FDA approved drugs, and the mechanisms are still not fully elucidated. It is actually maybe even more of the exception than the rule that you have a extremely clear understanding of exactly how something's necessarily working.
yeah, there's a number of seizure meds that mechanism unknown, but we use them.
Yeah.
Cool. A question about the trial logistics. Do we need to stay at the location for 12 weeks?
Addie, do you want to comment?
No, you do not. There will be 4 total visits. 2 of those will just be phone calls, and the other 2 will be in-person visits. So it's broken up such that the first one is a phone call with the investigator to go over the informed consent. The second one will be an in-person or in-clinic visit with the investigator. The third will be a phone call, and the fourth will be another in-person. So we do not anticipate anyone having to stay at the locations of the sites.
All right, thanks. I will take this question. This question says is for the Brain Foundation General. What other supplements are on the radar for ASD, specifically those with propensity for aggression to self and others? This is an area that needs a lot of attention as well. I just want to say we could not agree more with you. And in fact, it's very important for us to hear inputs like this from our community. A couple of months ago, that was the reason we sent out a survey to all our subscribers to tell us what are the most important areas of research and pain points where they need answers. If you did not get them, if you're not part of that, if you did not get the survey, please subscribe to our newsletter. Please follow us on Facebook and or LinkedIn so that you can be, so you are able to provide information like this. But in response to the specific questions, we are seeking proposals, and this is one of the areas that has been highlighted. In fact, the call for funding grants went out just this morning, and this is an area that we have asked researchers to apply with a proposal for. So hopefully we will have, we will be hosting Synchrony Symposium in October, where we will cover research on CBD, which can help with aggression, but there's a lot more that's needed than that. Other questions, I see a whole bunch of questions about access to the trial outside the US, access to the trial for adults. And I just want to reiterate again what was just said. The trial is for US participants, US residents only at this time. And hopefully the product will become available after in some time. And again, we hope to also be able to do another study for adults or non-speaking children as a follow-up to this trial. So please help us fundraise for that. If we are able to raise enough funds, that study will happen. Other questions, does Emodin address the symptoms of anxiety and aggression in children or teens?
So I think we're still, the purpose of this trial in a large way is to understand, more specifically, the symptoms that this can address. What we've seen, at least, as Ashley outlined in the presentation, is some potential benefits on behaviors, certain types of behaviors and memory, in particular related to sociability, and so it is possible that there can be some effects related to things like aggression. The answer is the purpose of the trial is really going to be to try and understand that better. We certainly hope there are, and I think there's the potential to, based on some of the initial research that has been done, but that's really the purpose of the work that we're undertaking here.
Okay, thanks. There's a question around European regulations. Zach, if you want to maybe just speak to that live. I see a lot of back and forth going on chat on this.
Yeah, I'm trying to keep up with both here.
Yes, so, I know. I'll give you some time to look at the question or just, speak what you know about the European regulations and the approval of Emodin.
Yeah, so I made one response, and if there's been other responses, I apologize. There's really quite a lot of activity, which is great to see. And then, of course, we can follow up as well if there's additional questions. Just please send them to Sarika, and we can get back to you. We're not intimately familiar with all of the regulations in Europe. We are aware of some of the activity.
Can you just restate the question, sorry, Zach, just for the people who will be listening to the recording?
Yeah. So let me find the question here. So I believe the question you're referring to is.
It's about Emodin,
yeah. It says by Dave Gaines, it says Emodin has recently been banned as a supplement in Europe due to concerns of it causing cancer and DNA damage. Can you address this in regard to your product? The response that I had written, and maybe I can just briefly elaborate on, is that we're not intimately familiar with the European regulations. We're under the impression that this was considered as really a class of compounds, somewhat related to aloe, which of course is something taken in a number of different ways across the world for a long period of time, in this particular class of compounds that come from that. I think we're using a lot of the word Emodin a lot, that the source of that, which I agree, it's not necessarily based on the source of where Emodin comes from. Obviously, Emodin is Emodin as from a compound, but this is a supplement product, meaning that it is not delivered as 100% pure Emodin. It's a extract of Japanese knotweed. It's an herbal extract, and of course we're using the highest quality sources, and have stayed well within the windows that we know where there were potential toxicities of the compound. So we've stayed well within those windows. The product, Japanese knotweed supplements as well as Piperine supplements are of course broadly available in the US. We're not aware of any similar concerns that FDA has expressed relative to what is being referenced here in Europe. So I do take the additional comment that I think was made in the comment box that there's been a lot of products from FDA that at one point were allowed and then shown to be toxic. I mean, and that's an obviously accurate statement. It's very hard to argue with something like that. Our knowledge of these compounds in the space is continually evolving. And I think we're, of course, going in a direction of just getting better and better understandings. I think all we can say with respect to this is, our product is a supplement product derived from herbal ingredients that are well within the guidelines that are allowed by FDA. We're not aware of concerns of the product being used in the way that we are outlining here. And, we'll obviously continue to monitor the situation. And if it seems that new evidence comes up that might indicate something, of course, we'll have to reevaluate certain things, but it's certainly not something that has raised concern from the individuals and scientists and regulators that we have spoken to about this.
Okay,
if that answers the question. Yeah.
Yeah, I'm sure there's a lot of back and forth, and it's impossible in a forum like this to, just continue further on that. But thank you so much. I think what we will do is collect these questions and make them available to PoloMar Health, and we will publish, an FAQ with answers to as many questions here. I see a lot of questions with similar themes, but we'll just try to get that out to you all, because there's just, I think it's going to be tough to go through all of the other questions as well in this forum at this time. What I'd like to do is walk people through just sort of what the key things that I'd like you all to hear from us, and I'm sharing a presentation here. One of the key things that is coming up for us is Synchrony in October at the Asilomar Conference Grounds. Please join us for that, to participate and listen into the roundtables we'll have, to give your patient input, to meet leaders of biopharma industries, and to hear, kind of the next, spate of research that we're pushing for around use of drugs that can be repurposed for autism. But as a final call to action regarding Neuromodin, if your child meets the trial inclusion or exclusion criteria, apply to participate at the link mentioned on the screen. It's brainfoundation.org/Neuromodin. Subscribe to our newsletter for similar updates on other similar opportunities. This is one. We hope that there'll be many more. Go to brainfoundation.org, scroll to the bottom of the page, and you can subscribe there. And finally, support our mission in whatever way you can. I think a lot of us wish for so many things to happen, but it can only happen if we as a community join hands and really participate in pushing this agenda that we want to come out. So join us as a volunteer. We are a complete volunteer-led organization, a team that's comprised of people from the autism community. Many of us, including me, have children we care for on the side, but we also try to make time to make this work happen. And every little bit that we can get in terms of help from you all will make a huge difference to what else we can do. Help spread awareness about our work and our events, and help us raise funds. We are very, very passionate about wanting to fund this trial for adults and for some of the other excluded subsets. That can happen. And again, because we're a public charity, we need funds to be able to make that happen, but we hope that will also come through soon because of that. So with that, I don't know if any of the other panelists have anything else to add. Let me open the floor up in case anybody has. Yes.
I just wanted to thank everybody for taking their time out on a Saturday to learn more about the product and the work that we're doing. I want to thank the Brain Foundation for their continued support and dedication to this space, and of course the investigators for all of their help in helping to design and understand the best way to approach this type of trial, and to my colleagues as well, and to all of you that are helping to advance research in this area. It's something we as well are very passionate about, and appreciate everybody taking the time.
Thank you, Zach.
Yeah, I think the company is to be commended for doing the study, because obviously it will give us more knowledge about whether or not the treatment works, as opposed to just releasing it to the public, right? So at least we'll have some data and know what it does, how it works, hopefully, or at least what symptoms it helps with. So I'm glad to see the study being done.
Absolutely. And, most supplements really have very little evidence supporting them often. So we're very excited that there will be, more evidence coming in the weeks or months to come on Neuromodin.
Right.
Well, with that, if there are no other words to add, thank you everyone for joining us today. We will make a recording of this event available to all registered attendees, and hope to see you at one of our future events. And thank you to the panelists.
Thank you.
Thank you, everyone.
Thank you.